Now we are going to the next section, which is types of Tox21 data mining of external data sources. So the learning objectives for this section are as follows. After attending this section, you should be able to define what is the difference between high throughput and high content data, and what are typical data sources of that, why data mining is important in Tox21, and you should be able to name some important direct and indirect sources for Tox21 data and what kind of data they contain. So let's start with types of Tox21 data. There are in principle three types of data. You can have high content, high throughput, but also reference data, reference classifications. Let's start with the third one, reference data classifications. These are normally results from traditional toxicological tests, animals in vitro tests, and these are the reference standard that you need to interpret the results from your high content or high throughput data. High content data is typically coming from imaging experiments, from arrays, or from omics experiments, and as the name already says, they have a high content, which means they are high dimensional. That means a lot of variables are measured in parallel. High throughput, on the other hand, are characterized by fast screening of libraries with a limited amount of tests and readouts. That means you only have a handful of readouts and tests, or even less, maybe just one, but you screen libraries which can have sizes of 1,000, 10,000, or even 100,000. So I just touched the name omics, and you have already learned in the lesson from Dr. Merkens some of the omics. I would like to come back a little bit to that. If most people talk about omics technologies, they think first about transcriptomics data, because almost every lab is able to measure that. But please be aware that transcriptomics only gives you a part of the picture. So on the right hand of this slide, you can see how complicated the interaction between a stressor and the ligand, the activation of transcription factors and kinases, and the feedback mechanisms of that could be. And that is why it is important not just to look at the expression level of genes. You also need as well to do genomics experiments. You maybe also need to go into epigenetics and measuring the methylation of DNA. But it is also important to go maybe into proteomics and to have a look at the phosphorylation of enzymes, so whether they are switched on and off. If you want to understand the metabolism, it is also maybe important to do metabolomics experiments, and never underestimate, and I said it already in the beginning, the power of visualization tools. Also to complement your results, and I will come back to that on one of the next slides, is text mining of external results. And all this together you can use in systems networking approaches to understand your data in a better way, to form hypotheses and validate hypotheses in there. But as toxicologists, we are not only just doing experiments by ourselves, there is a huge amount of publicly data which is available and can be assessed and used by us. So various direct and indirect sources of Tox21 or different data can and should be used. So this is the area of data mining. So how is data mining defined? Data mining is the practice of examining large databases in order to generate new information, hypotheses, or verify hypotheses in these data which you have generated elsewhere. So now I would like to talk about some typical data sources that you can use in your day-to-day scientific work. One of the already now pretty old ones is the so-called OECD QSAR toolbox, originally developed to fill in gaps in toxicity data needed to do hazard assessment of chemicals, but also for grouping of chemicals and read-across approaches. But It is a very powerful tool because OECD started many years ago to implement an extensive amount of worldwide toxicity databases from the different national or international regulators. And it is freely available. You can download it, and it's a very valuable source of data that you can use for Tox21 approaches. The second one, pretty famous in the United States, is the Toxcast dashboard. And you have already learned a lot of the details in the lecture of Dr. Kleinsteuer, which is modeling of hazard and organ manifestation. And the Toxcast dashboard has been developed as an interactive tool for the distribution, visualization, and the use of chemical screening data, especially in the context of the Toxcast project of the Tox21 collaboration. There's another very important source of data which is available since a few years, which is coming from ECHA, the European Chemical Agency. Since a few years, the European Union has REACH, which is the registration, evaluation, authorization, and restriction of chemicals regulation. And as part of this regulation, the manufacturers or importers are obliged to publish their testing results in the ECHA database, which means you have traditional toxicity data for more than 120,000 chemicals available, and the results are very detailed and includes controls, exactly which protocols are used, and is a very valuable source of information. But do not forget about the various data sources and pathway repositories that have not been developed in the direct context of Tox21 data. So I would like to sketch a few very important ones which I use in my day-to-day work. So GEO, the Gene Expression Omnibus from NIH. This is a publicly available database of genomics data, mainly array data, but also comparative genomic hybridizations or other kind of data in that area. And you can download the entire data sets there and can data mine them for things you are interested. Pretty famous pathway databases are WikiPathways, KEGG, and BioSig. WikiPathways is freely available. For KEGG and BioSig, you have, if you want to use the entire libraries and want to download, then you have to pay for them. But they have web interfaces where you can freely insert single searches. So if you just have a minor amount of questions that you would like to ask, you can go to the web page and access the databases there. Also, very important to understand what your genes are doing. Gene cards, a searchable integrative database that provides comprehensive user-friendly information on all annotated and predicted human genes. We talked a little bit in the last 10 minutes about Tox21 data sources and types of data.